Alright, nerd turds, here's what we're going to do. We are going to take about 2 weeks worth of organic chemistry and we're going to look for an underlying theme, and that theme would be condensation reactions. We're going to learn some condensation reactions today, and in the process we're going to summarize a whole bunch of what we would have done in Chem 1. A condensation reaction is a reaction in which two organic molecules are combined together and make one larger molecule, and as a result usually a water molecule is also created. It could also be another small molecule, but for all the molecules that we're going to look at, the extra molecule is going to be water. The first particular condensation reaction we're going to look at is called an esterification reaction. And in esterification, we make an ester. We use an alcohol and a carboxylic acid and react them together to make an ester and a water molecule. Now, esters are organic molecules that very often have strong and very specific scents, like a fruity scent in many cases. We do a lab in class where we make esters. Let's pick an alcohol for our example. Let's use ethanol. So we have a 2-carbon chain with an OH, a hydroxyl group on the right side here. This is our alcohol. And then we're going to pick a small carboxylic acid. Let's use propanoic acid for our carboxylic acid. Here is the acid part, and then a 3-carbon chain, carboxylic acid. Yeah. So what we're going to do is we are going to take the OH off of the alcohol and the H off of the acid and use it to make a water and mush together everything that's left over. Watch, this is how this goes. So here's our alcohol. We take the OH off of the alcohol and we use that connection spot to the carbon to hook it up to the oxygen of the propanoic acid. So again, I'll say that again and again here. We take the OH off the alcohol and the H off of the acid, and we use that to make water. And then the ester that's left is ethyl propanoate. Let's look at the models. Here's a model for ethanol, and there's propanoic acid. Again, what we're going to do is we are going to pull the OH off of the acid. We are going to pull the H off of the alcohol, and we're going to join those together to make a water molecule. There it is. And then we are going to attach everything that's left, mush it together, boom, ethyl propanoate. This example of condensation reactions is with protein synthesis. Now, proteins have a wide variety of functions in a living organism. One might say that you are a pile of proteins with a pulse, and what it is amino acids that are linked together into a chain. It's really a biopolymer. So what we need is 2 different amino acids, and we link them together to produce what biologists call a dipeptide, or what we would just call a diamino acid or a dimer in chemistry, and then there's water left behind as well. Let's look at amino acids. It turns out there are 20 different kinds of amino acids in all the living things on Earth, and they all are more similar than they are different. It's a 2-carbon chain with an amine on one side and a carboxylic acid on the other side. That's why they're called amino acids. It makes sense. What makes them different is what's called the side chain, and this one has got a side chain with just this hydrogen. It's the simplest of all the amino acids. It's called glycine. Let's pick another amino acid to join together with it. We'll use cysteine because it's pretty simple and won't take up too much space. Again, it starts off with 2 carbons attached to each other, and on one side we need to put an amine, and on the other side we're going to put a carboxylic acid. The side chain for a cysteine is more complicated. Alright, here we go. A cysteine has got a single carbon that's attached to a sulfur that's attached to a hydrogen. That sulfur hydrogen group is called a thiol. And this is the amino acid cysteine. So what we're going to do is we're going to join them together. We're going to pull the OH off of the acid and the H off of the amine in this case. It has a similar setup to what we saw with our esters, where we pulled the OH off the acid and the H off the alcohol. So here we go. We've got our glycine. What we're going to do is we're going to pop the OH off there. Now we're going to attach that carbon straight onto the nitrogen in cysteine, and this is what biologists like to call a peptide linkage. I don't know where biologists come up with these words like peptide. I've got some ideas that involve late nights in the lab with medicinal plants, but they don't make a lot of sense. The chemistry words make a whole lot more. So this is a dipeptide in biology, or a dimer, a diamino acid in chemistry. We would call this a glycine cysteine dimer. Let's look at the models. here's glycine with just hydrogen for a side chain, and here is cysteine. Cysteine has got that carbon connected to a sulfur, connected to a hydrogen. The sulfur is the yellow atom here. I'll try to arrange it so it looks like the drawing as much as possible. We're going to pull the OH off of the acid end of the one amino acid and the H off of the amine on the other end of the second amino acid, join them together to make a water molecule. And now we're going to join the carbon on the one to the nitrogen on the other, and we have two amino acids linked together. It's the beginning of a protein. The next important molecule for us to talk about are carbohydrates. In fact, complex carbohydrates are made by joining together simpler carbohydrates into chains using condensation reactions. We would call these simple sugars, or again, in biology, they would call these things monosaccharides. I have no idea what a saccharide is, but a simple sugar is joined together with another simple sugar to make a more complex carbohydrate, and you guessed it, water. Now, simple sugars are either 5 or 6 atom rings. We're going to start with glucose here. Glucose is a 6-atom ring. There's an oxygen, so it's what we call an ether ring because it's 5 carbons with an oxygen, so that makes an ether in a ring structure. And these structures are just bristling with OH groups. So every carbon in this molecule has got an alcohol or a hydroxyl group attached to it. And then we have to fill in the rest of the carbons with hydrogens. For our next simple sugar, let's use fructose. Fructose is a 5-atom ring. There is an ether ring. It's got that oxygen interrupting the carbon ring, and it's got a bunch of OH groups and other little dangly things hanging off onto the side of fructose. Like every other one of our condensation reactions, we're going to make a water molecule here, but it's slightly different in that there is no acid to take the OH off of. So we're going to take an OH off of one of the hydroxyl groups on the fructose and an H off of the glucose and use that to make a water. Now, when we mush what's left over, these two rings come together and they are joined together by what is called an ether linkage. You'll see when we get this all together what I mean, because we will have a carbon on one ring connected to an oxygen connected to a carbon on the next ring, and that carbon oxygen carbon is what we call an ether. This is the molecule sucrose. This is table sugar. Sweet. Let's go to the models. There is the model for glucose, and here is the model for fructose. What we're going to do is pull a particular OH group off of the fructose and then a hydrogen off of a particular spot on the glucose and put them together to make a water, and then mush together everything that's left over. If you end up having to draw this again, you'll have to refer back to this video, to your notes, or find something online. I don't expect that you're going to remember it from looking at this. And for our last trick, we're going to make a triglyceride. Now, triglycerides are the molecules that store energy in your fat cells. They also can be found in your bloodstream, but you don't really want them there. Ask your dad about his triglycerides. He's not going to be happy. So what a fatty acid is a long alkane chain with a carboxylic acid on the end. So it's really long, between 12 and 18 or 20 carbons long is how long that chain is. And we actually need 3 fatty acids to make a triglyceride. We're just going to use this zigzag line to show that it's a bunch of carbons linked together, and we'll assume that there's hydrogens on there as well to save ourselves some time and trouble. The other molecule we need is a molecule called 1,2,3-propatriol, would be the IUPAC name, otherwise known as glycerol. So it's a 3-carbon chain, and there is a OH, an alcohol group, on each of the carbons in the chain. Dots on there, and then we have to fill in the carbon's valence with hydrogens. What we're going to do is stick with the theme of pulling an OH off of each of the acids and an H off of the alcohols and joining them together into a package of 3 long chain fatty acids connected with a glycerol molecule. And in the process, we are going to make 3 water molecules. There they are. 3 long chain fatty acid molecules. Look at these guys. You guys look fatty. So there's a long chain. I think that's 14 carbons with a carboxylic acid on the end. That's 16 carbons, maybe 18 carbons. I think that's what I made. Here is our 1,2,3 propatriol. It's also called glycerol. We're going to pull the OH off the acid and H off of one of the alcohols and make a water molecule. And we're going to attach the acid onto the glycerol at that spot. Pull the OH off the acid. Pull the H off of the alcohol. I feel like I'm getting in a pattern here. Make another water molecule. And pull the OH off of the acid. Pull the H off of the alcohol. And make your third water molecule. Join up those acids onto the glycerol molecule at each of those locations, and you have got yourself a triglyceride, an energy storing molecule found in fat cells.